What's up, little biochemist? I'm Archen Suwan.
Hi, I'm John Cyril Villan.
And I am Alina Jane Aguilar.
And you're watching a Jack Biochemistry channel. For today's video, you will learn about the plasma membrane and the membrane transport.
Come on and join us in this amazing world of biochemistry.
We probably know that living organisms are made of cells. From unicellular organisms like bacteria, which are made of just one cell, to large animals that are made of trillions of cells. One key feature of cell is the cell membrane or plasma membrane, which separates what's inside the cell from what's outside of the cell. This is very important function because without the separation, there could be no such thing as life, because an organism must be distinct from its surroundings. Inside the cell is where metabolism takes place and genetic information is stored under regulated conditions of homeostasis. The cell membrane is semi-permeable, meaning it lets something through while preventing other things. The structure of plasma membrane is similar to the soap micelles, where the molecules arrange themselves with the hydrophilic heads facing out and hydrophobic tails pointing in, except that instead of a micelle, it is a phospholipid bilayer that engulfs the contents of the cells. It's called a bilayer because it's two layers of phospholipids which each have a phosphate group similar to the carboxylate group in the soap molecules, which formal charges that can interact with water molecules, and nonpolar fatty acid tails, one saturated, the other unsaturated, which prefer to stay hidden from water molecules. One layer faces outwards to the aqueous environment outside of the cell, and the other layer faces inwards to the aqueous environment inside the cell. That means there are two layers of nonpolar materials in between. This is part of what makes this membrane semi-permeable. Some compounds are able to traverse this nonpolar section and others are not. We refer to this bilayer as a fluid mosaic because these phospholipids are not fixed in their location with respect to one another. They are swimming around and constantly changing places like concert goers navigating to the crowd. Membrane transport is essential for cellular life. As cells proceed through their life cycle, a vast amount of exchange is necessary to maintain function. Transport may involve the incorporation of biological molecules and the discharge of waste products that are necessary for normal function. Moreover, the inner part of the cell is called the cytosol, and the outside of the cell is surrounded by a fluid called interstitial fluid. Substances move into and out of a cell through several different processes called membrane transport. There are two main processes: passive transport processes and active transport processes. The main difference between the two is that passive processes do not require energy expenditure, and active processes do require cells to expend energy. Let's start by looking at passive processes, which include simple diffusion, facilitated diffusion, and osmosis. Diffusion is the movement of a substance from where it has a high concentration to where it has a low concentration, or the tendency of a substance to spread out evenly over a given space. For instance, when a sugar cube is dissolved in water, over time, the cube will dissolve and eventually spread out evenly in the water until it reaches equilibrium. Diffusion occurs down a concentration gradient, which is a difference in concentration of a substance between two areas. So, the sugar molecules will move from an area of high concentration to an area of low concentration. Cellular diffusion is when diffusion of a solute, which is a dissolved substance, occurs across the plasma membrane from an area of high concentration to an area of low concentration. This is dependent on the concentration of a substance in the interstitial fluid outside the cell and the cytosol inside the cell. This can occur to simple diffusion or facilitated diffusion. Simple diffusion occurs with solutes that are small and nonpolar. By being nonpolar, they can move in between the phospholipid molecules that form the plasma membrane because the interior region of the membrane is nonpolar. Some of the materials that move by simple diffusion include the gases O2, CO2, and small fatty acids. So, if there is a higher concentration of oxygen molecules outside of a cell, they can move down the concentration gradient across the membrane without assistance and into the cell as long as the concentration gradient exists. And if there is a higher concentration of carbon dioxide molecules inside the cell, they can move across the membrane without assistance out of the cell into the interstitial fluid. So again, simple diffusion is when nonpolar molecules pass directly through a membrane.
The second type of diffusion is facilitated diffusion that applies to solutes that are small and either charged or polar. Because these solutes are polar, the nonpolar phospholipid bilayer blocks them from passing through the membrane and into or out of the cell by simple diffusion. However, they can pass into and out of the cell with the assistance of plasma membrane proteins through a process called facilitated diffusion. These are two types of facilitated diffusion: the channel-mediated diffusion and carrier-mediated diffusion. The difference between the two is the type of transport protein used to move the substance across the membrane. Channel-mediated diffusion is when an ion, which is a charged particle, where its total number of electrons does not equal its total number of protons, giving it a positive or negative charge, moves across the membrane through a water-filled protein channel. Each protein channel is typically specific for one type of ion, and there are two types of channels: a leak channel, which is continuously open, and a gated channel, which only opens due to a stimulus and only stays open for a fraction of a second. So, for a sodium positive ion, it can pass through a sodium positive leak channel continuously, and a gated sodium positive channel will only open due to stimulus to allow to open or to pass through into the cell. Channel-mediated diffusion involves the movement of polar molecules such as simple sugars or simple carbohydrates and amino acids across the membrane. This is accomplished by a carrier proteins, which actually changes shape in the process. For instance, glucose binds to a carrier protein, which changes shape and moves the glucose molecule to the other side of the membrane. Now, for osmosis, osmosis is the positive movement of water through a selectively permeable membrane. This occurs when there is a difference in concentration of water on either side of the membrane. This can happen in one of two ways. Water can slip between the phospholipid molecules that make up the plasma membrane, or through the integral part water channels that are called aquaporins. The plasma membrane is not permeable to most solutes such as charge, polar, and large substances. So example, one side of the membrane, the cytosol or interstitial fluid, can have more solutes than the other side. Let's say that the interstitial fluid has 3% solutes and 97% water, and the cytosol side has 1% solute and 99% cytosol side to the 97% interstitial side to achieve equilibrium. Now, let's look at active processes require the use of cellular energy for membrane transport. These are two types of active processes, active transport and vesicular transport. First, we have active transport. It is the movement of a solute against its concentration gradient or going from an area of low concentration to a place of higher concentration. While vesicular transport is the transport of large substances across the plasma membrane by a vesicle, which is a membrane-bound sac filled with materials. Active transport has two types, the primary active transport and the secondary active transport. In primary active transport, cellular protein pumps called ion pumps. It is move ion across the membrane against their concentration gradient. Let's take a look at this example. Here we have a membrane along with a sodium positive and potassium positive protein pump. 3 sodium ions and ATP bind the pump. ATP being like fuel or the form of cellular energy. In order to release that energy, ATP breaks down into ADP and P. P binds to the pump and the release of energy causes the pump to change shape and release the sodium positive ions into the interstitial fluid. 2 positive potassium ions bind to the pump and the P produced by the ATP is released into the cytosol of the cell. The transport protein or pump reverts back to its original shape, releasing the positive potassium ions into the cytosol. The pump is ready to do its job again.
In secondary transport, a substance is moved against its concentration gradient by using energy provided by the movement of second substance down its concentration gradient. So, a substance that is moving from a place of low concentration to high concentration. There are two types of secondary active transports. One is symport, where two substances are moved in the same direction, and antiport, where the two substances are moved in a different or opposite directions. In our example here, we have a positive sodium ions moving from a high concentration inside of the cell. A symporter will use the energy created by this to move a glucose molecule from a low concentration area to a high concentration area. And here, we have an antiporter again using energy from moving positive sodium ions from high concentration inside of the cell. And H positive is moved up its gradient and outside of the cell. Bisector transport involves the transport of a larger substance such as proteins or large carbohydrates across the plasma membrane. There are two types of vesicular transport. One is exocytosis and two is the endocytosis. In exocytosis, materials are secreted from the cell to the interstitial fluid outside of the cell. Typically, membranous vesicle formed by the Golgi apparatus transport materials to the cell membrane. At the membrane, the phospholipid molecules that makes up vesicle fuse with the phospholipid molecules that make up the plasma membrane, and the contents of the vesicle are released outside the cell. The vesicle membrane now becomes part of the plasma membrane. In endocytosis, the plasma membrane kinds of traps a substance by folding inwards. The lipids biolayer then fuse to form a vesicle surrounding the substance. There are three main forms of endocytosis. First is a phagocytosis, second is a pinocytosis, and for the last one is the receptor-mediated endocytosis. In Phagocytosis, a large particle is engulfed by the newly formed vesicle that contains digestive enzymes that breaks down the particles into its components molecules. And also phagocytosis is also called as cell eating. Penocytosis, which is also called the cell drinking, is when the plasma membrane folds inwards and engulfs droplets of interstitial fluid that contains dissolved solutes that can be used by the cell. Receptor-mediated endocytosis involves using receptors on the outside of the plasma membrane. These receptors binds with molecules in the interstitial fluids and the membrane's folds enclosing the receptors and the bound molecules to form a vesicle for transport within the cell. Remember both vesicle transport processes, endocytosis and exocytosis, requires energy.
That would be all for today's video.
We hope that you have learned new knowledge about the membrane transport.
Don't forget to like this video and subscribe to our channel for more videos like this.
See you next time, little biochemists. Bye!